Welcome to the second part of the lecture on the microbial network of nitrogen-converting microorganisms. Here, we will start from nitrate, the most oxidized form of nitrogen, and we will discuss the major microbial pathways catalyzing its reduction. First, besides ammonia, nitrate itself can serve as nitrogen source for biomass synthesis. This is termed assimilatory nitrate reduction. From a catabolic perspective, nitrate is reduced back to nitrite for energy production. Nitrate reduction, termed denitratation, occurs in all ecosystems where oxygen is absent, anoxic, or present at very low concentrations. This metabolic trait is widely spread across bacteria and archaea, and often nitrate reducers are facultative aerobes. Namely, they can also use oxygen as electron acceptor. Most nitrate reducers are heterotrophs, yet both organic and inorganic reduced compounds can serve as electron donors. Here, COD represents organic compounds. The produced nitrite sits at the crossroad of at least three different metabolisms, namely denitritation, DNRA, and Anamox. We will now discuss the ecological niche of the organisms performing each process. Denitritation involves the reduction of nitrite to dinitrogen gas via nitric oxide, NO, and nitrous oxide, N2O, as obligate intermediates. Organisms performing denitrifications are ubiquitous, primarily belonging to alpha, beta, and gamma proteobacteria classes, and are facultative aerobes. Most denitrifiers are heterotrophs, using organic carbon as carbon source. Importantly, denitrification is a modular path and often performed by a consortia of microorganisms catalyzing one or few steps instead of one performing the full pathway. Imbalances between the individual reduction rates may lead to the emission of N2O, which is a greenhouse gas 300 times stronger than CO2. In natural and engineered ecosystems, denitrifiers reducing nitrite to N2 compete with microorganisms reducing nitrite to ammonium for energy generation. This reduction is coupled to the oxidation of inorganic or typically organic electron donors. Ammonifiers are functionally diverse and can be facultative or obligate anaerobes and are predominantly heterotrophs. Among bacteria, ammonifiers are found in most phyla, with the four dominant ones reported on the slide. Ecologically, many factors contribute to the control the competition between denitrifiers and DNRA organisms. One widely recognized factor is the ratio between organic electron donors and nitrite or nitrate, where several lines of evidence suggest DNRA organisms to often feature higher nitrate affinity and nitrite affinity, in that they can thrive at lower residual concentrations. The third main pathway competing for nitrite is Anamox, anaerobic ammonium oxidation. Discovered in the early 90s, as we will discuss in the Anamox application class, Anamox bacteria denitrify nitrite to N2 with ammonium as electron donor. On the other side, they oxidize ammonium with nitrite as electron acceptor, a discovery that challenged the long-standing idea that oxygen is required to activate ammonium. Anamox bacteria belong to 5 main genera within the phylum Planktomycetes. They are autotrophs, and nitrite, the catabolic electron acceptor, serves as electron donor to fix CO2 into biomass. So Anamox typically produce a small amount of nitrite nitrate. Distinctive feature of Anamox bacteria is a membrane-bound intracellular compartment, the anamoxosome, where the catabolic reactions take place. The anamoxosome confines the highly reactive intermediate hydrazine, N2H4, a rocket fuel, a compound unique to the Anamox metabolism. Owing to their complex metabolism, Anamox are slow growers and therefore can compete with denitrifying and DNRA bacteria only in environments where ammonium and nitrite coexist and where oxygen and readily biodegradable COD are low. In conclusion, along the two classes about microbial nitrogen conversions, we learned about the main microorganisms that catalyze nitrogen turnover, their physiology and environmental niches. Nitrification, comprising nitritation and nitratation, is the first step in nitrogen removal in conventional wastewater treatment plants, while it leads to nitrogen loss from agriculture via nitrite and nitrate leaching to the surrounding water bodies. Similarly, denitrification, comprising denitratation and denitritation, coupled to nitrification, completes the removal of nitrogen in wastewater treatment plants. In turn, in agriculture, denitrification further contributes to the loss of nitrogen via volatilization, including the potential emissions of the potent greenhouse gas nitrous oxide. Conversely, DNRA is an unwanted process in wastewater treatment plant, while it retains nitrogen in agricultural soils. Ultimately, Anamox bacteria are key to the development of energy and resource-efficient wastewater treatment plants, as we will learn in the application class.